So I'm going to talk a little bit about drug development today. How many chemists do we have in the audience? Some chemists, that's good, so you can help me out here. And I'm going to talk about novel anti-cancer HSP90 inhibitors, and the project was to develop these. And see if this works? Yes, so first of all, some terminology. And do we have a...
The top one is the laser.
Oh yeah, good. So we're going to be talking today about quinones and hydroquinones, and you can see the difference. And I'll just introduce a few terms so we know what we're talking about as we move on here. You can see that the quinone moves to a hydroquinone by the addition of 2 electrons and 2 protons. We'll also be talking about an enzyme called NQO1, which Rick mentioned in the introduction. This is a metabolic enzyme, and it converts quinones to hydroquinones, the same reaction. So there's the quinone going to hydroquinone. NQO1 just drives it that way. And finally, we'll be talking about HSP90, which was in the title, and this is a promising target for drug development. So, by way of outline for this talk, I'll tell you why HSP90 is a good anti-cancer target. I'll talk to you about current quinone-based HSP90 inhibitors, the current compounds in clinical trial and clinical usage to inhibit HSP90 are quinones. You've just seen the structure of a typical quinone, benzoquinone. The structure of these molecules are a bit more heroic. They're benzoquinone ansamycins, but I'll show you that structure and take you through that. And finally, I want to talk about how we think we've exploited this metabolic enzyme, NQO1, in the development of novel HSP90 inhibitors. So that was the outline. So I'll give you the bottom line now, so in case you want to fall asleep during the rest of this talk, if you've had a hard, rough day in the classroom. The hydroquinone ansamycins that are generated via NQO1, mediated metabolism, are more potent HSP90 inhibitors than the parent quinones. So if we look at these structures, and I told you these structures were heroic. This is the yellow is the ansamycin portion, but remember the simple quinone that we talked about before in white, going to the hydroquinone, NQO1 catalyzing the reaction. It's these things that are the active agents, and we think we can utilize that observation in development of new HSP90 inhibitors. So, a little bit about what HSP90 actually is. HSP90 is a chaperone protein. It's expressed at high levels in most solid tumors. That's basically non-leukemias. What HSP90 does, what a chaperone protein does, it's critical for the folding and stability of a number of oncogenic client proteins. And what do I mean by that? Well, if you have an oncogenic protein which is driving neoplasia, it's coming off the ribosome, immediately being synthesized. It's not folded into its final effective conformation. It needs to be folded to do its job. It needs to be in the correct structure to do its job. And HSP90 is a chaperone. It's basically an ATPase that uses energy from cleaving ATP to fold proteins correctly. It's a protein catalyst, basically. And so you'll get correct folding of a number of these oncogenic proteins, and I've listed a few of them here. There are many of them. Some of those are the usual suspects people in oncology will look at, but the major point is they all drive the neoplastic process. Now, HSP90 doesn't fold only neoplastic oncogenic driving proteins. It folds many proteins, but if you look down the list, there are a lot of proteins that drive neoplasia in the category of HSP90 client proteins. And so that's why this is thought to be an interesting target. So if you want to inhibit the neoplastic process, if you want to inhibit cancer here, being driven by these things, where are you going to hit? Where are you going to design a molecule against? You're going to try and come in at the top of the schematic, so that if you do inhibit there, this doesn't show up too well, it's in red, if you inhibit here, then you get this combinatorial inhibition of a variety of different pathways. And that's the reason it's a pretty attractive drug target right now, and there's a lot of research and a lot of money going into this at the university and the company level. So the current benzoquinone ansamycin HSP90 inhibitors, there's the quinone again. Galvanomycin was the prototypical benzoquinone ansamycin. They all differ here at this R group. It's basically the only difference. So galvanomycin was a methoxy. There are other derivatives, 17-AAG and 17-DMAG, which are in clinical trials right now. And these have different substitutions simply at the 17 position. Okay? A little bit about NQO1 before we get into this. As I said, it's an enzyme. It's an obligatory 2-electron reductase which catalyzes quinones to hydroquinones. That's why it's called a quinone reductase. I just wanted to make the point that it's not causally involved in the pathway to neoplasia, not like HSP90 is, for example. NQO1, however, is elevated in human solid tumors. The reasons we don't really know why. We think it's, it's probably more of a stress response. Tumors are under stress. They have a high growth fraction. Environmental conditions, they're hypoxic. They're using a lot of, they're generating a lot of acids. It's probably elevated as a stress response. And to show you that, this will show. Yeah, this is some expression of this enzyme. It's immunohistochemistry. The brown staining is NQO1. And so you can see there's a fair amount of NQO1 expressed in all these different types of solid tumor. Thyroid, adrenal, breast, ovarian, colon, corneal tumor. We haven't put lung on there. And we were particularly interested to find recently, this is work we just published, that it's also highly expressed in pancreatic tumors. Here you see a human pancreatic tumor staining for NQO1, the brown staining, not much in the surrounding tissue. You also see that this is a metastatic pancreatic cancer that's gone into a lymph node, and you also see NQO1 picking up the metastatic tumor cells here. So NQO1 is elevated in these human tumors. So is there a role for NQO1 in the mechanism of action and metabolism of these HSP90 inhibitors? Now you already know that there is, because I've already given you the bottom line, so I wouldn't be presenting this if there wasn't. But you can see that the quinone going to a hydroquinone, and the first question we asked is, can NQO1 actually catalyze this reaction? Very simple question. And the answer is yes, it can. So NQO1 can affect this metabolism, but that sort of puts the hydroquinone at the scene of the crime, as Sherlock Holmes might say, but it doesn't really prove that the hydroquinone is committing the evil deed. We know it's being generated in these tumors by NQO1, but is it inhibiting HSP90? And I apologize for that slide, I just pulled it out quickly this morning. But so to try and define a role for the hydroquinone and benzoquinone in the HSP90 inhibition, we took 3 approaches. The first approach was an obvious one, to fish out the purified human proteins and look at the inhibitory actions of quinone and hydroquinone against those purified proteins, and actually use yeast and human HSP90. We did some molecular modeling of these benzoquinone ansamycins and their hydroquinone derivatives in the HSP90 active site, and I'll show you how we did that. And finally, we looked at some functional issues down the line. If you generate more of the hydroquinone in cells that overexpress NQO1, do you see more HSP90 inhibition and more growth inhibitory activity of these molecules? So just very briefly, this is looking at the first approach here, inhibition of yeast and human HSP90 by either the quinone, which is 17-AAG, or 17-AAG hydroquinone, which we generate through NQO1 metabolism. And just to take you to the important part of the slide, if you look at 17-AAG here, the quinone itself, with this particular concentration against yeast HSP90, we see very little inhibition of HSP90 activity. This is just by measuring its ATPase activity. We can we generate this hydroquinone, verify it's there. We generate it using this enzyme NQO1. We can also then block NQO1 with this molecule. I apologize for the acronym, it's ES936. It just inhibits this enzyme. It inhibits generation of the hydroquinone, and you can see we abrogate that inhibition. And the situation is qualitatively similar using human HSP90. It's quantitatively a little different. At this concentration of quinone, it's actually having a bit more of an inhibitory effect. and in the yeast system. But still, with the hydroquinone, we see more inhibition, and we abrogate that when we block NQO1. What about molecular modeling? I don't know if this is going to show up too well, but the molecular modeling is done by downloading a crystal structure of the protein. This is done using silicon graphics, molecular modeling, molecular mechanics modeling. And the protein is shown in gray here. The yellow is basically the structures of the benzoquinone ansamycins. You see a quinone on the left and a hydroquinone on the right. And so you'll try and then dock, if you like, the structures into these proteins, and you'll do 1,000 iterations and find, or 10,000 iterations and find the most energetically favorable structure and look at the binding energies. And so basically the quinone has this degree of total interaction energy with the protein, and the more the negative number here is, the better the binding. And so all this says is that the hydroquinone is binding more actively in the active site of HSP90 than the quinone. And the reason this is because of hydrogen bonding. You're generating 2 OH groups, and you're getting more hydrogen bonding in the active site of the protein. It's quite straightforward. So that looked like the hydroquinone was actually inhibiting HSP90. We wanted to check this out in a cell model, and so we took a cell, first of all, that we knew was NQO1 null, that didn't have NQO1 because of the genetic polymorphism that we characterized in this cells about 10 years ago. This is a human breast cancer cell line. We could transfect in NQO1 using a simple neomycin selection process, neomycin resistance marker. And so basically you generate a stable knock-in of NQO1, cells with a lot of NQO1 and cells with no NQO1, and you can compare those in the same genetic background, basically. And we verified, I'm not going to show you that data, that in those cells we could see hydroquinone generation from these quinones, and it was only in the NQO1 knock-in cells that we saw that. We saw increased HSP90 inhibition, and we also saw increased amount of growth inhibition in these systems with NQO1 when you compare it to systems lacking NQO1. So it seemed that if the cells had NQO1, they were generating the hydroquinone, they were inhibiting HSP90 more efficiently, and you were getting more growth inhibition. So we felt that this was to carry the investigative analogy probably a little far, but we felt this was case closed, that the hydroquinone was the most effective HSP90 inhibitor, and because of that was definitely in trouble and was going directly to jail. But so the question then was, could we exploit that a little more? We think we've proven that the hydroquinone is the most active HSP90 inhibitor. We can design prodrugs of these hydroquinones, and they can be designed to yield the hydroquinone ansamycins directly after hydrolysis. I'll show you examples of that. And the nice part about this is some of these hydrolyzing enzymes, hydrolases and esterases, are elevated again in tumor cells. So that provides another potential targeting approach. And I'll just give you I don't know if you can see that too well, but here's the structure of the hydroquinone, again, that we were dealing with here. And I've put in yellow some of the derivatives that we can use here. For example, this one is a sulfate. And sulfatases are high in breast tumors. And so can we use this as a targeting mechanism to get at certain breast cancers? So this is just the straightforward mechanism where we have our derivatives coming into a cell, a tumor cell, and then hydrolyzed by the appropriate enzyme to generate the hydroquinone, which is giving you HSP90 inhibition and hopefully a growth inhibitory effect. So in conclusion, I think I'm close to time here. NQO1 metabolizes these quinones to their hydroquinone forms. Hydroquinone ansamycins are more efficient at HSP90 inhibition than those parent quinones. So we figured that the hydroquinones were the active agents. These high levels of NQO1 in human tumor cells really can be exploited, I think, for generating the active forms of these HSP90 inhibitory quinones, the hydroquinones, directly in these tumor cells. And if we get a second kind of targeting approach, we can develop hydroquinone prodrugs as a useful approach, I think, to deliver these active hydroquinones directly to specific tumor cells. And so with that, I'd just like to thank the people that did this work. Wencha Angur, who was not a PhD when this work started, but he now is. David Siegel, Jeej Kepper, and Phil Regan in the lab. Our chemistry collaborator at the University of Nottingham, UK. Some of the original work that stimulated this was funded through an NIH grant, but all of the work that you see here essentially has been funded by this Bioscience Development and Evaluation Grant, the 1360, the Proof of Concept Award, and we thank you and the legislators for allowing us to do that. So with that, can we take questions?
Mm-hmm. Yeah.
Any questions? Yes, sir.
When you say hydrolysis are elevated in tumors, by how much? What's the expression?
Yeah, usually you're looking at the sulfatases in breast cancer are about, tenfold and upwards. And, one does have to make sure that, the particular sulfatase that's going to do the cleavage of, let's say, a sulfate derivative, because there's a large family of sulfatases. You have to verify that that particular sulfatase will do that cleavage, of course. It's a good question. Yes, sir?
What does the enzyme usually do if there's no drug around? Why is it there?
That's a great question. And, we think it's there, again, as I mentioned earlier on, as a stress response. It will help scavenge superoxide, which is, a normal product of, free radical product of mitochondrial respiration. It'll generate antioxidant forms of quinones in the body. And we think, from some recent data, which actually is now published, I guess we can talk about it, that it does stabilize p53, which is a tumor suppressor gene. So it has a few different functions normally. And I actually like to think of, and if you look at the amount of this protein in cells, there's a lot of it, and so, I like to think that if cells are taking the trouble to make lots of a particular protein, they're going to have that protein doing many different jobs. So sort of a multitasking protein, which can do many different things depending on the environment and the stresses. But to answer your question in a nutshell, I think it's a stress response.